chimeric promoters for use in plants. Wo 2,005,069,986. To abstract, the present invention provides polynucleotide molecules useful for expressing transgenes in plants. The present invention also provides expression constructs containing the polynucleotide molecules useful for expressing transgenes in plants. The present invention also provides transgenic plants and seeds containing the polynucleotide molecules useful for expressing transgenes in plants. Claims. OCR text may contain errors. We claim 1. A chimeric promoter comprising a collamovirus promoter enhancer fused with a plant actin gene promoter to form said chimeric promoter. 2. The chimeric promoter of Claim 1 wherein said collamovirus promoter enhancer comprises a cauliflower mosaic virus 35S promoter enhancer. 3. The chimeric promoter of Claim 2 wherein said cauliflower mosaic virus 35S promoter enhancer comprises a nucleic acid sequence selected from the group consisting of seqid no. 2-6. 4. The chimeric promoter of Claim 1 wherein said plant actin gene promoter comprises an Arabidopsis actin gene promoter. 5. The chimeric promoter of Claim 4 wherein said Arabidopsis actin gene promoter is seqid no. 8. 6. The chimeric promoter of Claim 1 wherein said plant actin gene promoter comprises a rice actin gene promoter. 7. The chimeric promoter of Claim 6 wherein said rice actin gene promoter is seqid no. 7. 8. The chimeric promoter of Claim 1 wherein said chimeric promoter comprises a nucleic acid sequence selected from the group consisting of seqid no. 9-35. 9. The construct comprising a chimeric promoter comprising a collamovirus promoter enhancer fused with a plant actin gene promoter to form said chimeric promoter, wherein said chimeric promoter is operably linked to a transcribable polynucleotide molecule operably linked to a 3-transcription termination polynucleotide molecule. 10. The construct of Claim 9 wherein said chimeric promoter is selected from the group consisting of seqid no. 9-35. 11. The construct of Claim 9, wherein said transcribable polynucleotide molecule is a marker gene. 12. The construct of Claim 9, wherein said transcribable polynucleotide molecule is a gene of agronomic interest. 13. The DNA construct of Claim 12, wherein said gene of agronomic interest is a herbicide tolerance gene selected from the group consisting of genes that encode for phosphinothioacetyltransferase, glyphosate-resistant 5-enol pyruvyl shikimate 3, phosphate synthase, hydroxyphenylpyruvate dehydrogenase, valipon dehalogenase, bromoxynil-resistant nitrulase, anthranilate synthase, glyphosate oxidoreductase, dicambamonoxygenase, and glyphosate N-acetotransferase. 14. A transgenic plant stably transformed with a construct comprising a chimeric promoter comprising a collamovirus promoter enhancer fused with a plant actin gene promoter to form a chimeric promoter, wherein said chimeric promoter is operably linked to a transcribable polynucleotide molecule operably linked to a 3-transcription termination polynucleotide molecule. 15. A transgenic plant of Claim 14, wherein said transcribable polynucleotide molecule is a gene of agronomic interest. 16. A transgenic plant of Claim 14, wherein said transcribable polynucleotide molecule is a marker gene. 17. A seed of said transgenic plant of Claim 14. 18. The method of inhibiting weed growth in a field of transgenic glyphosate-tolerant crop plants comprising planting the transgenic plants transformed with an expression set comprising an isolated polynucleotide molecule having gene regulatory activity and comprising a polynucleotide sequence selected from the group consisting of seqid no. 9-35 and operably linked to a DNA molecule encoding a glyphosate tolerance gene and applying glyphosate to the field at an application rate that inhibits the growth of weeds, wherein the growth and yield of the transgenic crop plant is not substantially affected by the glyphosate application. 19. In the method of Claim 18, wherein said glyphosate tolerance gene is selected from the group consisting of a gene encoding for a glyphosate-resistant 5-enol pyruvyl shikimate 3-phosphate synthase, a glyphosate oxidoreductase, and a glyphosate N-acetyltransferase. 20. In the method of Claim 18, wherein the transgenic plants are capable of tolerating an application rate up to 256 ounces/acre. 21. In the method of Claim 18, wherein the transgenic plants are capable of tolerating an application rate ranging from 8 ounces/acre to 128 ounces/acre. 22 and the method of claim 18, wherein the transgenic plants are capable of tolerating an application rate ranging from 32 ounces/acre to 96 ounces/acre. 23 and the method of claim 18, wherein the application of glyphosate is at least once during the growth of the crop. Description: OCR text may contain errors. Chimeric promoters for use in plants. This application claims the benefit of U.S. application 60/537,793 filed January 28, 2004, incorporated herein by reference in its entirety. Incorporation of sequence listing to copies of the sequence listing CQ. Listing copy 1 and CQ. Listing copy 2 and computer-readable form of the sequence listing, all on CD-ROMs, each containing the file name pot_01117.rpt, which is 61,440 bytes measured in MS-DOS and was created on January 18, 2005, all of which are incorporated herein by reference. Field of the invention: the invention relates to the field of plant molecular biology and plant genetic engineering and polynucleotide molecules useful for modulating gene expression in plants. Background: one of the goals of plant genetic engineering is to produce plants with agronomically desirable characteristics or traits. The proper expression of a desirable transgene in a transgenic plant is one way to achieve this goal. Promoters are non-coding polynucleotide molecules which play an integral role in the overall expression of genes in living cells. Isolated promoters that function in plants are useful for modifying plant phenotypes through the methods of genetic engineering. Many constitutive promoters are available and are useful for providing good overall gene expression. For example, constitutive promoters such as PFMB, the promoter from the 35S transcript of the Fig wart mosaic virus, U.S. patent number 6051753, page CAM35S, the promoter from the 35SR, the transcript of the cauliflower mosaic virus, U.S. patent number 5530196, P. rice actin-1, the promoter from the actin-1 gene of Oryzaeus sativa, U.S. patent number 5641876, and PNOS, the promoter from the no balance in face gene of Agrobacterium tumefaciens, are known to provide some level of gene expression in most or all of the tissues of a plant during most or all of the plant's lifespan. Alternately, many promoters are available with more specific expression patterns such as tissue specificity, temporal specificity, or developmental specificity. These promoters are useful for the targeted expression of a transgene in plants. Optimal expression of a transgene is useful for producing plants with agronomically desirable characteristics or traits. Such optimal expression often requires a promoter having a specific expression pattern which may not be readily available in known promoters. One example of such a specific expression pattern is a high level of transgene expression in both vegetative and reproductive tissues. The present invention solves this problem by producing novel chimeric promoters containing elements from known promoters. These novel chimeric promoters can then be tested in plants to determine whether the desired expression pattern is indeed achieved. Summary: in one embodiment, the invention provides novel chimeric promoters provided as SEQID No. 9-35, comprising a columnovirus promoter enhancer fused with a plant actin gene promoter and useful for modulating gene expression in plants. In another embodiment, the invention provides constructs comprising the novel chimeric promoter and useful for modulating gene expression in plants. In another embodiment, the invention provides a transgenic plant comprising the novel chimeric promoter and the seed of the transgenic plant. In another embodiment, the invention provides a method of inhibiting weed growth in a field of transgenic glyphosate-tolerant crop plants comprising planting the transgenic plants transformed with an expression set comprising the novel chimeric promoter operably linked to a DNA molecule encoding a glyphosate tolerance gene and applying glyphosate to the field at an application rate that inhibits the growth of weeds. Brief description of the drawings. Figure 1 represents a section of the CAMB35S promoter with enhancer domains marked. Also diagrammatically represented are 5 enhancer domains constructed for use in creating chimeric actin promoters. Figure 2 represents the native rice actin-1 promoter and novel chimeric promoters made by fusing the rice actin-1 promoter and selected CAMB35S promoter enhancer domains. Figure 3 represents the native Arabidopsis actin-1 promoter and novel chimeric promoters made by fusing the Arabidopsis actin-1 promoter and selected CAMB35S promoter enhancer domains. Detailed description. The following definitions and methods are provided to better define the present invention and to guide those of ordinary skill in the art in the practice of the present invention. Unless otherwise noted, terms are to be understood according to conventional usage by those of ordinary skill in the relevant art. The invention disclosed herein provides novel combinations of polynucleotide molecules for use in constructing novel chimeric promoters. The design, construction, and use of chimeric or hybrid promoters comprising one or more of the enhancer domains of a colimovirus 35S promoter and a plant actin gene promoter is one object of this invention. The novel chimeric promoter sequences thereof SEQID No. 9-35 are capable of transcribing operably linked DNA sequences in multiple tissues and therefore can selectively regulate expression of transgenes in multiple tissues. As used herein, the term polynucleotide molecule refers to the single, or double-stranded DNA or RNA of genomic or synthetic origin, i.e., a polymer of deoxyribonucleotide or ribonucleotide bases, respectively, read from the 5 upstream end to the 3 downstream end. As used herein, the term polynucleotide sequence refers to the sequence of a polynucleotide molecule. The nomenclature for DNA bases as set forth at 37 CFR 1.822 is used. As used herein, the term gene regulatory activity refers to the ability to affect transcription or translation of an operably linked transcribable polynucleotide molecule. An isolated polynucleotide molecule having gene regulatory activity may provide temporal or spatial expression or modulate levels and rates of expression of the operably linked transcribable polynucleotide molecule. An isolated polynucleotide molecule having gene regulatory activity may comprise a promoter, intron, leader, or 3 transcription termination region. As used herein, the term gene expression refers to the transcription of a DNA molecule into a transcribed RNA molecule. Gene expression may be described as related to temporal, spatial, developmental, or morphological qualities as well as quantitative or qualitative indications. As used herein, the term regulatory element refers to a polynucleotide molecule that may affect the transcription or translation of an operably linked transcribable polynucleotide molecule. Regulatory elements such as promoters, leaders, introns, and transcription termination regions are non-coding polynucleotide molecules having gene regulatory activity which play an integral part in the overall expression of genes in living cells. Isolated regulatory elements that function in plants are therefore useful for modifying plant phenotypes through the methods of genetic engineering. As used herein, the term promoter refers to a polynucleotide molecule that is involved in recognition and binding of RNA polymerase II and other proteins transacting transcription factors to initiate transcription. The plant promoter is a native or non-native promoter that is functional in plant cells. A promoter can be used as a 5 regulatory element for modulating expression of an operably linked transcribable polynucleotide molecule. Promoters may be defined by their temporal, spatial, or developmental expression pattern. As used herein, the term enhancer domain refers to a cis-acting transcriptional regulatory element, aka cis element, which confers an aspect of the overall control of gene expression. An enhancer domain may function to bind transcription factors, transacting protein factors that regulate transcription. Some enhancer domains bind more than one transcription factor, and transcription factors may interact with different affinities with more than one enhancer domain. Enhancer domains can be identified by a number of techniques, including deletion analysis, i.e., deleting one or more nucleotides from the 5' or internal to a promoter, DNA binding protein analysis using DNA footprinting, methylation interference, electrophoresis mobility shift assays, in vivo genomic footprinting by ligation-mediated PCR, and other conventional assays, or by DNA sequence similarity analysis with known cis element motifs by conventional DNA sequence comparison methods. The fine structure of an enhancer domain can be further studied by mutagenesis or substitution of one or more nucleotides or by other conventional methods. Enhancer domains can be obtained by chemical synthesis or by isolation from promoters that include such elements, and they can be synthesized with additional flanking nucleotides that contain useful restriction enzyme sites to facilitate subsequent manipulation. Thus, the design, construction, and use of enhancer domains according to the methods disclosed herein for modulating the expression of awkwardly linked transcribable polynucleotide molecules are encompassed by the present invention as used herein. The term chimeric refers to the product of the fusion of portions of two or more different polynucleotide molecules, as used herein. The term chimeric promoter refers to a promoter produced through the manipulation of known promoters or other polynucleotide molecules. Such chimeric promoters may combine enhancer domains that can confer or modulate gene expression from one or more promoters. For example, by fusing a heterologous enhancer domain from a first promoter to a second promoter with its own partial or complete regulatory elements. The novel chimeric promoters of the present invention desirably contain at least one enhancer domain fused to a plant actin promoter. Thus, the design, construction, and use of chimeric promoters according to the methods disclosed herein for modulating the expression of awkwardly linked transcribable polynucleotide molecules are encompassed by the present invention, as used herein. The term percent sequence identity refers to the percentage of identical nucleotides in a linear polynucleotide sequence of a reference query polynucleotide molecule or its complementary strand as compared to a test subject polynucleotide molecule or its complementary strand when the two sequences optimally aligned with appropriate nucleotide insertions, deletions, or gaps totaling less than 20% of the reference sequence over the window of comparison. Optimal alignment of sequences for aligning a comparison window are well known to those skilled in the art and may be conducted by tools such as the local homology algorithm of Smith and Waterman, the homology alignment algorithm of Needleman and Winch, the search for similarities method of Pearson and Littman, and preferably by computerized implementations of these algorithms such as GAPT, BSDFIT, FASTA, and TFASTA available as part of the GCG registered trademark Wisconsin package registered trademark Acceleris Incorporated, Burlington, MA. An identity fraction for aligned segments of a test sequence and a reference sequence is the number of identical components which are shared by the two aligned sequences divided by the total number of components in the reference sequence segment, i.e., the entire reference sequence for a smaller defined part of the reference sequence. Percent sequence identity is represented as the identity fraction multiplied by 100. The comparison of one or more polynucleotide sequences may be to a full-length polynucleotide sequence or a portion thereof, or to a longer polynucleotide sequence, as used herein. The term substantial percent sequence identity refers to a percent sequence identity of at least about 80% sequence identity, at least about 90% sequence identity, or even greater sequence identity, such as about 98% or about 99% sequence identity. Thus, one embodiment of the invention is a polynucleotide molecule that has at least about 80% sequence identity, at least about 90% sequence identity, or even greater sequence identity, such as about 98% or about 99% sequence identity with a polynucleotide sequence described herein. Polynucleotide molecules that are capable of regulating transcription of awkwardly linked transcribable polynucleotide molecules and have a substantial percent sequence identity to the polynucleotide sequences of the promoters provided herein are encompassed within the scope of this invention. Promoter isolation and modification methods. Any number of methods well known to those skilled in the art can be used to isolate fragments of a promoter disclosed herein. For example, PCR polymerase chain reaction technology can be used to amplify flanking regions from a genomic library of a plant using publicly available sequence information. A number of methods are known to those of skill in the art to amplify unknown polynucleotide molecules adjacent to a core region of known polynucleotide sequence. Methods include but are not limited to inverse PCR, iPCR, vector-rep PCR, Y-shaped PCR, and genome walking approaches. Polynucleotide fragments can also be obtained by other techniques such as by directly synthesizing the fragment by chemical means, as is commonly practiced by using an automated oligonucleotide synthesizer. For the present invention, the polynucleotide molecules were isolated by designing PCR primers based on available sequence information. Novel chimeric promoters can be designed or engineered by a number of methods. For example, a chimeric promoter may be produced by fusing an enhancer domain from a first promoter to a second promoter. The resultant chimeric promoter may have novel expression properties relative to the first or second promoters. Novel chimeric promoters can be constructed such that the enhancer domain from a first promoter is fused at the 5' end, at the 3' end, or at any position internal to the second promoter. The location of the enhancer domain fusion relative to the second promoter may cause the resultant chimeric promoter to have novel expression properties relative to a fusion made at a different location. Those of skill in the art are familiar with the standard resource materials that describe specific conditions and procedures for the construction, manipulation, and isolation of macromolecules, e.g., polynucleotide molecules, plasmids, etc., as well as the generation of recombinant organisms and the screening and isolation of polynucleotide molecules. Constructs as used herein. The term construct refers to any recombinant polynucleotide molecule such as a plasmid, cosmid, virus, autonomously replicating polynucleotide molecule, phage, or linear or circular single-stranded or double-stranded DNA or RNA polynucleotide molecule, derived from any source, capable of genomic integration or autonomous replication, comprising a polynucleotide molecule where one or more transcribable polynucleotide molecule has been operably linked. As used herein, the term operably linked refers to a first polynucleotide molecule, such as a promoter, connected with a second transcribable polynucleotide molecule, such as a gene of interest, where the polynucleotide molecules are so arranged that the first polynucleotide molecule affects the function of the second polynucleotide molecule. The two polynucleotide molecules may be part of a single contiguous polynucleotide molecule and may be adjacent. For example, a promoter is operably linked to a gene of interest if the promoter regulates immediate transcription of the gene of interest in a cell. As used herein, the term transcribable polynucleotide molecule refers to any polynucleotide molecule capable of being transcribed into a RNA molecule. Methods are known for introducing constructs into a cell in such a manner that the transcribable polynucleotide molecule is transcribed into a functional mRNA molecule that is translated and therefore expressed as a protein product. Constructs may also be constructed to be capable of expressing antisense RNA molecules, in order to inhibit translation of a specific RNA molecule of interest. For the practice of the present invention, conventional compositions and methods for preparing and using constructs and host cells are well known to one skilled in the art. See for example, Molecular Cloning, a Laboratory Manual, 3rd edition volumes 1, 2, and 3 2000. J. F. Sambrook, D. W. Russell, and N. Irwin, Cold Spring Harbor Laboratory Press. Constructs of the present invention would typically contain a promoter operably linked to a transcribable polynucleotide molecule operably linked to a 3-transcription termination polynucleotide molecule. In addition, constructs may include but are not limited to additional regulatory polynucleotide molecules from the 3-UNTR-ANSLA-TED region 3-UTR of plant genes, e.g., a 3-UTR to increase mRNA stability of the mRNA, such as the pi termination region of potato or the octafine or no balance in phase 3. Termination regions. Constructs may include but are not limited to the 5 untranslated regions 5-UTR of an mRNA polynucleotide molecule, which can play an important role in translation initiation and can also be a genetic component in a plant expression construct. For example, non-translated 5-liter polynucleotide molecules derived from heat shock protein genes have been demonstrated to enhance gene expression in plants. See for example, U.S. Patent Number 5,659,122 and U.S. Patent Number 5,362,865, all of which are incorporated herein by reference. These additional upstream and downstream regulatory polynucleotide molecules may be derived from a source that is native or heterologous with respect to the other elements present on the construct. Thus, one embodiment of the invention is a promoter such as provided in SEQID No. 9-35, operably linked to a transcribable polynucleotide molecule so as to direct transcription of said transcribable polynucleotide molecule at a desired level or in the desired tissue or developmental pattern upon introduction of said construct into a plant cell. In one embodiment, the transcribable polynucleotide molecule comprises a protein coding region of a gene, and the promoter provides for transcription of a functional mRNA molecule that is translated and expressed as a protein product. In another embodiment, The transcribable polynucleotide molecule comprises an antisense region of a gene, and the regulatory element affects the transcription of an antisense RNA molecule or other similar inhibitory RNA in order to inhibit expression of a specific RNA molecule of interest in a target host cell. Exemplary transcribable polynucleotide molecules for incorporation into constructs of the present invention include, for example, polynucleotide molecules or genes from a species other than the target species, or genes that originate with or are present in the same species, but are incorporated into recipient cells by genetic engineering methods rather than classical reproduction or breeding techniques. The type of polynucleotide molecule can include but is not limited to a polynucleotide molecule that is already present in the plant cell, a polynucleotide molecule from another plant, a polynucleotide molecule from a different organism, or a polynucleotide molecule generated externally, such as a polynucleotide molecule containing an antisense message of a gene, or a polynucleotide molecule encoding an artificial, synthetic, or otherwise modified version of a transgene. The regulatory elements of the present invention can be incorporated into a construct using marker genes as described and tested in transient or stable plant analyses to provide an indication of the regulatory element's gene expression pattern in stable transgenic plants. As used herein, the term marker gene refers to any transcribable polynucleotide molecule whose expression can be screened for or scored in some way. Marker genes for use in the practice of the present invention include, but are not limited to, transcribable polynucleotide molecules encoding glucuronidase GUS described in U.S. Patent Number 5,599,670, which is incorporated herein by reference, and green fluorescent protein GFP described in U.S. Patent Number 5,491,084 and U.S. Patent No. 6,146,826, both of which are incorporated herein by reference proteins that confer antibiotic resistance, or proteins that confer herbicide tolerance. Useful antibiotic resistance markers, including those encoding proteins conferring resistance to kanamycin and PELL hygromycin BFID streptomycin or spectorhidomycin AOD, spec/strep and gentamicin AAC3 and AACC4 are known in the art. Herbicides for which transgenic plant tolerance has been demonstrated and the method of the present invention can be applied, include but are not limited to glyphosate, glufosinate, sulfonylureas, imidazolinones, bromoxynil, Delapon, cyclohexanedione, protoporphyrinogenic oxidase inhibitors, and isoxyls-flutol herbicides. Polynucleotide molecules encoding proteins involved in herbicide tolerance are known in the art, and include, but are not limited to, a polynucleotide molecule encoding 5-enol pyruvylshikimate, 3-phosphate synthase PPSPs described in U.S. Patent Number 5,627,061. U.S. Patent Number 5,633,435, U.S. Patent Number 6,040,497 and in U.S. Patent Number 5,094,945 for glyphosate tolerance, all of which are incorporated herein by reference polynucleotides encoding a glyphosate oxidoreductase and a glyphosate N-acetyltransferase geodex described in U.S. Patent 5,463,175 and GAP described in U.S. Patent Publication 20,30,083,480, both of which are incorporated herein by reference a polynucleotide molecule encoding bromoxynil nitrulase B-accent described in U.S. Patent Number 4,810,648 for bromoxynil tolerance, which is incorporated herein by reference a polynucleotide molecule encoding phytoanddesaturase CRTL described in Misawa et al. 1993 Plant J4:833-840 and Misawa et al. 1994 Plant J6:481-489 for norflorazone tolerance, a polynucleotide molecule encoding acetohydroxyacid synthase. AKDALS described in Safajevan et al. 1990 NuCl. Acid stress. 18:2188-2193 for tolerance to sulfonylurea herbicides, a polynucleotide molecule encoding a dicamba-degrading oxygenase enzyme described in U.S. patent publications U.S. 20,30,135,879 and U.S. 20,30,115,626 for dicamba tolerance, all of which are incorporated herein by reference and the bar gene described in Dedlock, et al. 1987 EMBOJ 6:2513-2519 for glufosinate and bialafose tolerance. The regulatory elements of the present invention can express transcribable polynucleotide molecules that encode for phosphonothrysin acetyltransferase, glyphosate-resistant EPSPs, aminoglycoside phosphotransferase, hydroxyphenylpyruvate dehydrogenase, hydromycin phosphotransferase, neomycin phosphotransferase, valipon dehalogenase, bromoxynil-resistant nitrulase, anthranilate synthase, glyphosate oxidoreductase and glyphosate N-acetotransferase. Thus, in one embodiment of the invention, a polynucleotide molecule of the present invention as shown in SEQID No. 9-35 is incorporated into a DNA construct such that a polynucleotide molecule of the present invention is operably linked to a transcribable polynucleotide molecule that provides for a selectable, screenable, or scorable marker. The constructs containing the regulatory elements operably linked to a marker gene may be delivered to the tissues and the tissues analyzed by the appropriate mechanism, depending on the marker. The quantitative or qualitative analyses are used as a tool to evaluate the potential expression profile of a regulatory element when operatively linked to a gene of agronomic interest in stable plants. Any marker gene can be used in a transient assay. Methods of testing for marker gene expression and transient assays are known to those of skill in the art. Transient expression of marker genes has been reported using a variety of plants, tissues, and DNA delivery systems. For example, types of transient analyses can include but are not limited to direct gene delivery via electroporation or particle bombardment of tissues in any transient plant assay using any plant species of interest. Such transient systems would include but are not limited to electroporation of protoplasts from a variety of tissue sources or particle bombardment of specific tissues of interest. The present invention encompasses the use of any transient expression system to evaluate regulatory elements operably linked to any transcribable polynucleotide molecule, including but not limited to marker genes or genes of agronomic interest. Examples of plant tissues envisioned to test intransience via an appropriate delivery system would include but are not limited to leaf-based tissues, callus, cotyledons, roots, endosperm, embryos, floral tissue, pollen, and epidermal tissue. In one embodiment of the invention, a polynucleotide molecule as shown in SEQID No. 9-35 is incorporated into a construct such that a polynucleotide molecule of the present invention is operably linked to a transcribable polynucleotide molecule that is a gene of agronomic interest. As used herein, the term gene of agronomic interest refers to a transcribable polynucleotide molecule that includes but is not limited to a gene that provides a desirable characteristic associated with plant morphology, physiology, growth and development, yield, nutritional enhancement, disease or pest resistance, or environmental or chemical tolerance. The expression of a gene of agronomic interest is desirable in order to confer an agronomically important trait. The gene of agronomic interest that provides a beneficial agronomic trait to crop plants may be, for example, including, but not limited to genetic elements comprising herbicide resistance. U.S. Patent 6,803,501, 6,448,476, 6,248,876, 6,225,114, 6,107,549, 5,866,775, 5,804,425, 5,633,435, 5,463,175, and U.S. Patent Publications U.S. 20,30,135,879 and U.S. 20,30,115,626 increased yield U.S. patents. UFRE 38,446, 6,716,474, 6,663,906, 6,476,295, 6,441,277, 6,423,828, 6,399,330, 6,372,211, 6,235,971, 6,222,098, 5,716,837. Insect Control U.S. Patent 6,809,078, 6,713,063, 6,686,452, 6,657,046, 6,645,497, 6,642,030, 6,639,054, 6,620,988, 6,593,293, 6,555,655, 6,538,109, 6,537,756, 6,521,442, 6,501,009, 6,468,523, 6,326,351, 6,313,378, 6,284,949, 6,281,016, 6,248,536, 6,242,241, 6,221,649, 6,177,615, 6,156,573, 6,153,814, 6,110,464, 6,093,695, 6,063,756, 6,063,597, 6,023,013, 5,959,091, 5,942,664, 5,942,658, 5,880,275, 5,763,245, 5,763,241. Fungal disease resistance U.S. Patent 6,653,280, 6,573,361, 6,506,962, 6,316,407, 6,215,048, 5,516,671, 5,773,696, 6,121,436, 6,316,407, 6,506,962. Virus resistance U.S. Patent 6,617,496, 6,608,241, 6,015,940, 6,013,864, 5,850,023, 5,304,730. Nematode resistance U.S. Patent 6,228,992. Bacterial disease resistance U.S. Patent 5,516,671. Plant growth and development U.S. Patent 6,723,897. 6,518,488. Starch production U.S. Patent 6,538,181. 6,538,179, 6,538,178, 5,750,876, 6,476,295. Modified oils production U.S. Patent 6,444,876, 6,426,447, 6,380,462. High oil production U.S. Patent 6,495,739, 5,608,149, 6,483,008, 6,476,295 Modified fatty acid content U.S. Patent 6,828,475, 6,822,141, 6,770,465, 6,706,950, 6,660,849, 6,596,538, 6,589,767, 6,537,750, 6,489,461, 6,459,018 High protein production U.S. Patent 6,380,466 Fruit ripening U.S. Patent 5,512,466 Enhance animal and human nutrition U.S. Patent 6,723,837, 6,653,530, 65,412.59, 5,985,605, 6,171,640 Biopolymers U.S. Patents USRE 37,543, 6,228,623, 5,958,745 and U.S. Patent Publication No. U.S. 20,030,028,917 Environmental Stress Resistance U.S. Patent 6,072,103 Pharmaceutical Peptides and Secretable Peptides U.S. Patent 6,812,379, 6,774,283, 6,140,075, 6,080,560 Improved processing traits U.S. Patent 6,476,295 Improved digestibility U.S. Patent 6,531,648 Low raffinose U.S. Patent 6,166,292 Industrial enzyme production U.S. Patent 5,543,576 Improved flavor U.S. Patent 6,011 199 Nitrogen fixation U.S. Patent 5229, 114 Hybrid seed production U.S. Patent 5,689,041 Fiber production U.S. Patent 6,576,818, 6,271,443, 5,981,834, 5,869,720 and biofuel production U.S. Patent 5,998,700 The genetic elements, methods, and transgenes described in the patents listed above are incorporated herein by reference. Alternatively, a transcribable polynucleotide molecule can affect the above-mentioned plant characteristic or phenotypes by encoding the RNA molecule that causes the targeted inhibition of expression of an endogenous gene, for example via antisense, inhibitory RNA suppression-mediated mechanisms. The RNA could also be a catalytic RNA molecule, i.e., the ribozyme engineered to cleave a desired endogenous mRNA product. Thus, any transcribable polynucleotide molecule that encodes a transcribed RNA molecule that affects the phenotype or morphology change of interest may be useful for the practice of the present invention. The constructs of the present invention are generally double-T plasmid border DNA constructs that have the right border RB or A gram to .RB and left border pounds or A gram tux B regions of the T plasmid isolated from Agrobacterium tumefaciens comprising a tDNA, which along with transfer molecules provided by the Agrobacterium cells, permit the integration of the tDNA into the genome of a plant cell. The constructs may also contain the plasmid backbone DNA segments that provide replication function and antibiotic selection in bacterial cells. For example, an Escherichia coli origin of replication such as OR/322, a broad host range origin of replication such as OR/OR3, and a coding region for a selectable marker such as SPEC/STRP that encodes for TN7 aminoglycoside adenyltransferase A conferring resistance to spectinomycin or streptomycin, or a gentamicin gram, gent-selectable marker gene. For plant transformation, the host bacterial strain is often Agrobacterium tumefaciens ABI, C58, or LBA 4404. However, Other strains known to those skilled in the art of plant transformation can function in the present invention. Transformed plants and plant cells is used herein. The term transformed refers to a cell, tissue, organ, or organism into which has been introduced a foreign polynucleotide molecule, such as a construct. The introduced polynucleotide molecule may be integrated into the genomic DNA of the recipient cell, tissue, organ, or organism such that the introduced polynucleotide molecule is inherited by subsequent progeny. A transgenic or transformed cell or organism also includes progeny of the cell or organism and progeny produced from a breeding program employing such a transgenic plant as a parent in a cross and exhibiting an altered phenotype resulting from the presence of a foreign polynucleotide molecule. The plant transformation construct containing a promoter of the present invention may be introduced into plants by any plant transformation method. Methods and materials for transforming plants by introducing the plant expression construct into a plant genome in the practice of this invention can include any of the well-known and demonstrated methods including electroporation as illustrated in U.S. Patent Number 5384253, microprojectile bombardment as illustrated in U.S. Patent NOS 5015580, U.S. Patent Number 5550318, U.S. Patent Number 5538880, U.S. Patent Number 6160208, U.S. Patent Number 6399861, and U.S. Patent Number 6403865. Agrobacterium Hediated transformation as illustrated in U.S. Patent Number 5824877, U.S. Patent Number 5591616, U.S. Patent Number 5981840, and U.S. Patent Number 6384301, and protoplasm transformation as illustrated in U.S. Patent Number 5508. 184, all of which are incorporated herein by reference. Methods for specifically transforming dicots are well known to those skilled in the art. Transformation and plant regeneration using these methods have been described for a number of crops including, but not limited to, cotton Gossypium hirsutum, soybean Glycine max, peanut Ericus hypogaea, and members of the genus Brassica. Methods for transforming monocots are well known to those skilled in the art. Transformation and plant regeneration using these methods have been described for a number of crops including, but not limited to, barley Hordeum vulgare, maize Zira, maize oats Avena sativa, orchard grass Dactylis glomerata, rice Oryzea sativa, including indica and japonica varieties, sorghum bicolor, sugarcane Saccharomyces petal, Fescue festuca, rondinaceae or turf grass species e.g. species, Agrostis stolonifera, Poprotensis, Stenotaphyrum secundavium, wheat Triticum aestivum, and Alfalfa medicago sativa. It is apparent to those of skill in the art that a number of transformation methodologies can be used and modified for production of stable transgenic plants from any number of target crops of interest. The transformed plants are analyzed for the presence of the genes of interest and the expression level and / or profile confirmed by the promoters of the present invention. Those of skill in the art are aware of the numerous methods available for the analysis of transformed plants. For example, methods for plant analysis include, but are not limited to southern blots or northern blots, PCR-based approaches, biochemical analyses, phenotypic screening methods, field evaluations, and immunodiagnostic assays. The seeds of this invention can be harvested from fertile transgenic plants and be used to grow progeny generations of transformed plants of this invention, including hybrid plant lines comprising the construct of this invention and expressing the gene of agronomic interest. The present invention also provides for parts of the plants of the present invention. Plant parts, without limitation, include seed, endosperm, ovule and pollen. In a particularly preferred embodiment of the present invention, the plant part is a seed. Still yet another aspect of the invention is a method of inhibiting weed growth in a field of transgenic crop plants, comprising first planting the transgenic plants transformed with an expression set comprising a polynucleotide sequence selected from the group consisting of SEQID No. 9 to 35 operably linked to 8. transcribable polynucleotide molecule encoding a glyphosate tolerance gene and then applying glyphosate to the field A** when application rate that inhibits the growth of weeds, wherein the growth and yield of the transgenic crop plant is not substantially affected by the glyphosate application. The glyphosate application rate is the effective rate necessary to control weeds in a particular glyphosate-tolerant crop. These rates may range from 8 ounces/acre to 256 ounces/acre, preferably 16 ounces/acre, to 128 ounces/acre, and more preferably 32 ounces/acre to 96 ounces/acre. The glyphosate is applied at least once during the growth of the glyphosate-tolerant crop and may be applied 2, 3, or 4 times during the growth of the crop or more as necessary to control weeds in the field. The following examples are included to demonstrate preferred embodiments of the invention. It should be appreciated by those of skill in the art that the techniques disclosed in the examples that follow represent techniques discovered by the inventors to function well in the practice of the invention. However, those of skill in the art should, in rigidity of the present disclosure, appreciate that many changes can be made in the specific embodiments that are disclosed and still obtain a like or similar result without departing from the spirit and scope of the invention. Therefore all matter set forth or shown in the accompanying drawings is to be interpreted as illustrative and not in a limiting sense. Examples: Example 1. Construction of chimeric promoters. Novel chimeric promoters are constructed by fusing at least one enhancer domain from a columbovirus promoter with a plant actin gene promoter. A brief description of the sequences referred to herein is provided in Table 1 below. Table 1. Brief listing of key SEQID node figure IMGF 000017_0001 figure IMGF 000018_0001 figure IMGF 000019_0001. The columbovirus promoter can be any promoter from a virus in the columbovirus family, including but not limited to promoters from cauliflower mosaic virus can be such as the promoter from the 35S RNA transcript of the cauliflower mosaic virus PCAMB.35S SEQID node 1 U.S. patent number 5530, 196 and promoters from figwort mosaic virus FMV such as the promoter from the 35S transcript of the figwort mosaic virus, U.S. patent number 6,051,753, all of which are incorporated herein by reference. Promoter enhancer domains constructed as multimers of enhancer domains from the PCAM.35S promoter include but are not limited to the 4xB3 domain SEQID node, 2 4xA cell SEQID node, 3 2xBLB2 domain SEQID node, 4 2ZLB3 domain SEQID node, 5 and 2xBLB5 domain SEQID node, 6. See Figure 1. The monomers comprising the multimers listed above as well as other enhancer domains from PCAM.35S are described in U.S. patent number 5,097,025, incorporated herein by reference. The plant actin gene promoter can be any promoter from a plant actin gene, including but not limited to actin promoters from Oryzea sativa such as the rice actin-1 promoter POS.ACT-EL-SEQID node, 7 U.S. patent number 5,641,876 actin promoters from Arabidopsis thaliana such as the Arabidopsis actin-1 promoter P.ACT-EL-SEQID node, 8 and actin promoters from Xeramase such as the actin-2 promoter U.S. patent number 6,670,467, all of which are incorporated herein by reference. The fusion of at least one enhancer domain with a plant actin gene promoter may be to any region of the plant actin gene promoter including but not limited to the 5 end of the plant actin gene promoter, the 3 end of the plant actin gene promoter, or any region internal to the plant actin gene promoter. The enhancer domain may be in either the reverse or the forward orientation. Enhancer domains derived from PCAM.35S were inserted in 3 locations in the rice actin-1 relative to rice actin-O transcription initiation site -848 NT Hindle site -462 NT Ecury site and -80 NT Efsel site. See Figure 2. Construction of these chimeric promoters is described in detail below. The P4XB3/POS.ACTL chimeric promoters were created by fusing the 4 tandem copies CAM-B35SB3 enhancer sequence to the RICE-ACTL promoter POS.ACTL. Chimeras were made by fusing the enhancer region at the -848 NT position of POS.ACTL SEQID node 9 at the -462 NT position of POS.ACTL SEQ ID no. 10 and at the -80 NT position of POS.ACTL SEQID node 11. The P4XA cell/POS.ACTL chimeric promoter was created by fusing 4 tandem copies of the CAM-B35S as one enhancer sequence to the RICE-ACTL promoter POS.ACTL. Chimeras were made by fusing the enhancer region at the -848 NT position of POS.ACTL SEQID node 12 at the -462 NT position of POS.ACTL SEQID node 13 and at the -80 NT position of POS.ACTL SEQID node 14. The P2XBLB2/POS ACTL chimeric promoter was created by fusing 2 tandem copies of the CAM-B35SB1B2 enhancer sequence to the RICE-ACTL promoter POS.ACTL. Chimeras were made by fusing the enhancer region at the -848 NT position of POS.ACTL SEQID node 15 at the -462 NT position of POS.ACTL SEQID node 16 and at the -80 NT position of POS.ACTL SEQID node 17. The P2XBLB3/POS.ACTL chimeric promoters were created by fusing 2 tandem copies of the CAM-B35SA1B3 enhancer sequence to the RICE-ACTL promoter POS.ACTL. Chimeras were made by fusing the enhancer region at the -848 NT position of POS.ACTL SEQID node 18 at the -462 NT position of POS.ACTL SEQID node 19 and at the -80 NT position of POS.ACTL SEQID node 20. The P2XB1B5 POS ACTL chimeric promoter was created by fusing 2 tandem copies of the CAM-B35SB1B5 enhancer sequence to the RICE-ACTL promoter POS.ACTL. Chimeras were made by fusing the enhancer region at the -848 NT position of POS.ACTL SEQID node 21 at the -462 NT position of POS.ACTL SEQID node 22 and at the -80 NT position of POS.ACTL SEQID node 23. Enhancer domains derived from PCAM.35s were inserted in 4 locations in the Arabidopsis Actin1 promoter PAT.ACTL with the PAT.ACTL transcription initiation site designated as +1. 4 unique restriction sites in PAT.ACTL were used as insertion sites: Averil, BeastC, SIL, BSMV. Construction of these chimeric promoters is described in detail below. The 2 ZALD3 and 4XA cell fragments were isolated by restriction enzyme digest. Fragments ends were then made blunt by treatment with the 4D polymerase. Vectors containing the PAT.ACTL promoter were cut with one of the 4 unique enzymes and then the fragment ends were made blunt. Each of the 2 enhancer domains was fused into each of the 4 blended restriction enzyme sites. Chimeric promoters were selected with inserts in both the reverse and forward orientations. See Figure 3. Selected chimeric promoters were subcloned into a binary vector to operatively link the chimeric promoter with a reporter gene such as the GUS reporter gene J3-glucuronidase gene or the CP4 gene for glyphosate tolerance. These vectors were used for plant transformation and subsequent promoter characterization. Transgene expression levels of the chimeric promoters were compared with the transgene expression levels of control constructs. Example 2. Promoter characterization in transient systems. Selected chimeric promoters were used for transient transformation for reporter expression analysis. Transient systems used included tobacco protoplasts, corn protoplasts, wheat ovary, wheat anther, and barley microspores. Cells were harvested and protein extracted for GUS activity analysis. Methods for measuring GUS activity are well known to those skilled in the art. See for instance using the GUS gene as a reporter of gene expression 1992 edited by Sean R. Gallagher, Academic Press, Incorporated, San Diego. Monocot analysis constructs containing chimeric POS.ActL promoters were used to transform COM protoclasts and assay for Gus/Lux activity relative to the POS.ActL promoter and the PCAM.E35S promoter. Data are provided in Table 2. Table 2. Transient analysis in corn protoclasts figure IMGF 00022_0001. All the chimeric promoters tested were found to have higher activity than the native POS.ActL promoter when tested in COM protoclasts. The three chimeric promoters P-Tazol B3/P, OS.ActL3 SEQID No. 20P for XACell/POS.ActL3 SEQID No. 14 and P for XB3 POS.ActL 3 SEQID No. 11 also showed increased activity when compared to the CAM-E35S promoter. Two factors contributed the enhanced activity in the chimeric promoter, namely the enhancer domain selected and the fusion location of the enhancer domain. Every enhancer domain tested appeared to be more powerful when the fusion location was closer to the 3 end of the promoter, and attenuated when the fusion location was at the 5 end of the promoter. Among the enhancer domains tested, the Tazol B3 enhancer was found to be the strongest enhancer domain in COM protoclasts. The 4XACell enhancer was found to convey desirable strength to the chimeric promoter. The 4XB3 enhancer was found to be most effective when fused closer to the transcription start site. One limitation of expression analysis in the COM protoclast system is that it only represents vegetative tissue. An increased expression level in the COM protoclast system is not necessarily indicative of performance in reproductive tissue. A few selected chimeric promoters were therefore further tested for Gus activity PMAL/minimum in Lao in wheat and thir, wheat ovary, barley microspore, and COM pollen transient assay systems for comparison with COM leaf protoclast data. Data are provided in Table 3 below. In comparison to POS.ActL promoter, which is known to express well in reproductive tissue, these chimeric promoters did not show any decrease of activity in reproductive tissue. Table 3. Transient analysis of selected chimeric promoters in reproductive tissues figure IMGF 00023_0001 dicot analysis constructs containing chimeric P.ActL promoters were used to transform tobacco protoclasts and assay for Gus activity NHAM/G total protein. Data are provided in Table 4 below. Table 4. Transient analysis in tobacco protoclasts figure IMGF 00023_0002 to figure IMGF 00024_0001. Both the fusion location and choice of enhancer domain was found to produce a significant effect on promoter activity in tobacco protoclasts. The two promoters with the highest Gus activity were the P4XACell/@.ActL/NSEI No. 34 and P4XAs 1/@.ActL/B70 SEQID No. 35. For constructs comprising the Tazol B3 enhancer, the highest activity of Gus was shown with the enhancer fused at the B70 position. The Tazol B3 chimeric promoters were generally less active in all positions when compared to the 4XACell chimeric promoters. Reverse orientation of the enhancer domain did not change significantly the activity of the chimeric promoter. Example 3. Characterization of chimeric promoters in transgenic corn plants. Selected chimeric promoters were used for stable COM plant transformation for reporter expression analysis. Plants were transformed using agrobacterium-mediated methods. In order to have a direct side-by-side comparison of Gus activity in the cytoplasm and CP4 expression in plastids, and to minimize variations in sampling and environmental factors, a few selected chimeric promoter constructs were built. Each test construct comprised the test promoter driving the Gus reporter gene beta-glucuronidase coding sequence from E. coli and the test promoter driving the CP4 gene bacterial strain CP4-ARO gene encoding class IIBTSP's enzyme in a linear array. For test constructs, Pmin 46172 P-Tazol B3/POS.ActL3, SEQID No. 20 Pmin 46173 P4XACell/POS.ActL3, SEQID No. 14 Pmin 46174 P4XB3/POS.ActL2, SEQID No. 10 and Pmin 46175 Ptazl B3/POS.ACTL, SEQID no, 18 and the reference construct, Pmin 46170 PCAMD.E35S driving GUS and POS.ACTL driving CP4 were used to transform COM. Transformed COM plants were selected in glyphosate containing medium. 3R0 plants were generated for each transformation event. The first plant was sprayed with the equivalent of 64 ounce/acre of Roundup registered trademark Ultra. The second plant was sprayed with the equivalent of 96 ounce/acre of Roundup registered trademark Ultra. The third plant from each event was left as unsprayed control. 10 to 14 days after Roundup registered trademark application, each plant was rated for % chlorosis and % malformation. At mid-pollen shed, each RO plant was rated for male fertility. Positive RO plants were pollinated with LH198 pollen grains to produce FI seed. Transgenic events with single or lowest copy numbers of cassette based on CP4 copy number estimation by Tachman were selected for FI count analysis. The total of 5 events from each construct with triplicate plants in each data point were used. The plants were grown in greenhouse from FI seeds, and selected for positive segregates via glyphosate spray at 16 ounces/acre when plants reach Vita stage. Leaf tissue was taken at N4 stage, and V4 again at V8/V9 stage. Also at N8/V9 stage, tissue was collected from V8 leaf, root tip, and the mature tassel ranging from 0.5 cm to 3 cm. Pollen was collected when shedding. Embryo and endosperm were harvested at 12 days after pollination. Several positive embryos were pooled from GUS-positive individual kernels. Samples were extracted and used for both GUS quantitative analysis and CP4 ELISA. The Ptazl B3/POS.ACTL3 and P4XA/POS.ACTL3 chimeric promoters provided GUS gene expression which was as good as or better than that provided by PCAMD.E35S in most of the tissues analyzed. The P4XB3/POS.ACTL2 and Ptazl B3/POS.ACTL1 chimeric promoters provided high levels of expression in pollen with moderate or low levels of expression in other tissues as compared to that provided by PCAMD.E35S. The data are provided for GUS activity p-mol/minimum/milligrams protein as mean and standard error measurements in Table 5 below. Table 5. FI transgenic corn GUS activity p-mol/minimum/milligrams protein figure IMGF 00025_0001. All 4 chimeric promoters provided CP4 gene expression which was as good as or better than that provided by POS.ACTL in all of the tissues analyzed with the exception of pollen. Expression levels in pollen for chimeric promoter constructs were approximately 20% to 73% that of expression levels in pollen for POS.ACTL constructs. Data are provided for CP4 expression levels GCP4 protein/G total protein as mean and standard error measurements in Table 6 below. Table 6. FI transgenic corn CP4 expression levels GCP4 protein/G total protein figure IMGF 00026_0001. The FI progenies were also used for field tests. 3 FI populations derived from 3R0 plants of each event were tested. Commercial Roundup Ready registered trademark COM lines GA21 and NK603 were used as positive controls. 3 Roundup registered trademark rates were used in this test, 0, 96, and 128 ounces/A. Roundup registered trademark was applied at V4 leaf stage. Data obtained were % transformation efficiency TE number of single copy events per total events generated. Leaf CP4 levels GCP4/G fresh weight tissue pollen CP4 levels GCP4/G fresh weight tissue % chlorosis CHL % malformation male at 10 to 14 days after treatment and male fertility score MFR measured 1 to 5 with 5 the highest. Data represent the average score collected across the events generated for each construct and only tasseled plants were included for some measurements. Plants transformed with the P4XB3POS.ACTL2 or the P4XACLPOS.ACTL3 chimeric promoter constructs had chlorosis scores equivalent to the reference construct Pmin 46170. P4XACL/POS.ACTL3 had a malformation score lower than that of the reference construct. All chimeric promoters tested had male fertility rates comparable to that of plants transformed with the reference construct. Data are provided in Table 7 below. Table 7. Roundup registered trademark tolerance in transgenic cornfield tests. Figure IMGF 00027_0001. Example 4. Characterization of chimeric promoters in transgenic dicots. Several constructs were evaluated in transgenic Arabidopsis plants for GUS expression. GUS expression in leaf and flower tissue was measured as pm/mug/g total protein, and results were averaged for all the events produced for construct. The CAMB-E35S promoter P, CAMB-E35S and Arabidopsis actin-1 promoter P@.ActL were used as controls. Data are provided in Table 8 below. Table 8. Transgenic Arabidopsis analysis with chimeric that ActL promoters. Figure IMGF 00027_0000 to figure IMGF 00028_0001. GUS expression analysis in Arabidopsis showed that P-tazol B3/@.ActL/Beast CR/SEQID no 26 and P-tazol B3/@.ActL/BSEMB/SEQID no 29 provided transgene expression in flowers comparable to that of the P@.ActL promoter. The P-tazol B3/@.ActL/Beast CR/SEQID no 26 P for XACell/@.ActL/BSEMB/SEQID no 35 P-tazol B3/@.ActL/RBI/SEQID no 24 and P-tazol B3/@.ActL/BSEMB/SEQID no 29 promoters provided transgene expression in leaves higher than or comparable to the P@.ActEL and PCAMB.E35S. Example 4. Insect control analysis in corn to constructs PMO 38858 and PMIN 38859 were used to generate transgenic plants. Transformations were performed using agrobacterium-mediated methods. Both constructs contained the tazol B3/POS.ActL3 promoter SEQID no 20 operably linked to a nucleotide sequence in Cohenocry to AB insecticidal protein U.S. patent number 6489542. Several transgenic plant lines produced from each construct were analyzed for Cry to AB protein levels. The tazol B3/POS.ActL3 promoter was found to express the Cry2AB protein at high levels in COM leaf tissue when compared with standard controls. Example 5. Glyphosate tolerance analysis in wheat. 3 chimeric promoters were tested in transgenic wheat plants. Transgenic wheat plants were generated from each of the single cassette constructs PMIN 43646 P for XACell/P, OS.ActL3, SEQID no 14 PMIN 43647 P-tazol B3/POS.ActL2, SEQID no 19 and PMIN 43648 P-tazol B3/POS.ActL3, SEQID no 20. Transformations were performed with agrobacterium in immature white embryos. All 3 constructs contain a single copy of the CP4ETSP's gene for glyphosate tolerance. The distinguishing element in each construct is the promoter. Events from each single cassette construct were analyzed for vegetative and reproductive tolerance to glyphosate equivalent to the double cassette lead event 33391 U.S. patent publication US 20,020,062,503 generated from PMIN 30139 which contains 2 copies of the CP4ETSP's gene driven by the PE35S and POS ActL promoters, respectively. Plants were analyzed for glyphosate tolerance, phenotype, copy number, molecular profile, and genome location. Results are provided below. RO plants were spray tested for vegetative and reproductive tolerance with 64-A Roundup registered trademark Ultra 1.68 kg/Ha acid equivalents of glyphosate prior to jointing. Plants with vegetative damage were discarded. Fertility was estimated by counting the number of seeds in 20 florets from the central portion of the head and reported as % fertility. Data are provided in Table 9 below. Table 9. Glyphosate tolerance in transgenic RO wheat plants. Figure IMGF 000029_0001 data on the vegetative and reproductive glyphosate tolerance of R0 wheat plants transformed with a single cassette vector containing CP4 driven by the rice-actin promoter Pmin 30167 and a double cassette vector containing two CP4 genes driven by pCAM.E35S and POS.ACT-EL are also provided in Table 15. Wheat transformation experiments that used the pCAM.E35S cassette alone resulted in plants that were vegetatively tolerant but had low fertility. For example, plants generated with PMO 42411, comprising only the pCAM E35S promoter driving the CP4 gene, produced only one of 37 vegetatively tolerant plants with a fertility of greater than equals 80%. This data indicates that the E35S promoter is responsible for vegetative expression of CP4 while the rice-actin promoter is responsible for reproductive expression of CP4. Results for the single cassette vector containing the three chimeric promoters Pmin 43646, Pmin 43647, and Pmin 43648 were similar to those for the double cassette vector Pmin 30139. 73 to 75% of the total events generated using these three chimeric promoter constructs had vegetative tolerance, and 20 to 26% of those events had greater than equals 80% fertility. Thus, using one of the three chimeric promoters to drive a single copy of the CP4 ETSP's gene produced similar results in R0 tests as double cassette lead event. In addition to glyphosate tolerance, transformation efficiencies TE leaf CP4 levels GCP4 milligrams total protein from RO wheat plants and meristem CP4 levels GCP4 milligrams total protein from RO wheat plants were measured in RO plants. Plants transformed with Monday 30139 the double cassette construct and Pmin 30159 PSCDVC US patent number 6489462 were used as controls. Data are provided in Table 10 below. Table 10. CP4 expression in transgenic R0 wheat plants. Figure IMGF 000030_0001 RL seeds were then collected from the R0 plants and advanced to RL testing. RL plants were simultaneously tested for glyphosate tolerance and copy number. The transgene copy number for each event was determined by Southern blot and or Tachman quantitative PCR analysis. CP4 gene sequences were used as probes for both assays. A good correlation was observed between single copy calls on Southern's and by Tachman analysis. Single copy events, with good tolerance to high doses of glyphosate, were advanced to the R2 generation. Seeds from selected wheat lines advanced to the R2 generation were planted into pots and sprayed with 128 ounces/8 Roundup at the 3-leaf stage. Retained lines were transferred to larger pots and sprayed again with 128 ounces/8 at the 6-leaf stage. In an attempt to force differentiation among events, an extreme pressure test was devised where plants were sprayed with 512 ounces/8 of Roundup registered trademark. This was repeated 4 times between the 3-leaf stage and emergence of the flag leaf. Results for the chimeric promoter lines were compared with results for non-transgenic Bob White plants. R4 generation lead event plants line 33391 generated from Pmin 30139, and R3 generation plants containing the PSCB and promoter line TA_S2520 generated from PMO 30159. Yield datagrams and fertility data as % of total plants were collected for each line. Data are provided in Table 11 below. Table 11. Pressure test of R2 wheat plants. Figure IMGF 000030_000 to figure IMGF 000031_0001. From this analysis it is apparent that the use of the P4XA cell/POS.ACTL3 node 14, Petazole B3 POS.EDL to SEQID node 19, and Petazole B3/POS.ACTL3 SEQID node 20 promoters in plant CP4 expression cassettes confers glyphosate tolerance to vegetative tissues in wheat plants without negatively impacting fertility. In wheat, cassettes containing the rice-actin promoter without these elements produce plants with low vegetative tolerance and are not useful for the production of glyphosate-tolerant plants. Therefore, one advantage of the chimeric promoters in wheat is that vegetative and reproductive tolerance can be achieved without the use of a double CP4 cassette. Field trials were performed with ARFA generation plants to assess the performance of selected lines. Field trials were done at 8 U.S. sites with 4 replications each and treatments of 0, 64, and 128 ounces/A Roundup registered trademark ultra equivalent. Each treatment was arranged as a separate, randomized complete block in order to collect equivalence data on unsprayed non-transformed bobwhite plants. All events had vegetative and reproductive tolerance comparable to the lead event 33,391 at doses up to 128 ounces/A of Roundup registered trademark ultra. Yield was measured in tons/hectare T/ha. Data are provided in Table 12 below. Table 12, yield data from field trials of ARFA plants T/ha figure IMGF 00031_0002 having illustrated and described the principles of the present invention. It should be apparent to persons skilled in the art that the invention can be modified in arrangement and detail without departing from such principles. We claim all modifications that are within the spirit and scope of the appended claims. All publications and published patent documents cited in this specification are incorporated herein by reference to the same extent as if each individual publication or patent application is specifically and individually indicated to be incorporated herein by reference.